Welcome to Green Dolphin Science, and thank you for joining us for this deep dive. Today, we're looking at something you probably see around your home or workplace all the time, but have likely never thought of as a potential scientific tool: spiderwebs.
That's right. We're diving into some really fascinating research, actually. It explores whether these intricate structures can act as sort of silent monitors for a growing concern in our indoor environments, specifically microplastics and the chemical hitchhikers they often carry.
Yeah, we've got a study here titled Spiderwebs as Passive Monitors of Microplastic and Its Co-Pollutants in Indoor Environments. And our goal today is to really dig into this for you. We want to understand what this research tells us about the air we breathe indoors, the surprisingly effective role spiderwebs might play in capturing pollutants, and, what the implications could be for our health. Okay, so to start, what's the big problem this study is trying to address? Why look at indoor air?
Well, it's becoming increasingly clear that the air inside our buildings, where, let's face it, many of us spend the vast majority of our time, can actually be more polluted than the air outside. This is often because, indoor spaces just have less air circulation. Pollutants can build up.
Right, things get trapped. And one of the emerging villains in this indoor air quality story is microplastics, or as the study often calls them, MPs. We're talking about incredibly small pieces of plastic, less than 5 millimeters in size, like smaller than a grain of rice. And the study points out that things we have all around us indoors, like our clothes, textiles, single-use plastics, these are major sources.
Exactly. And what's particularly interesting, and frankly a bit concerning, is that the plastic itself isn't the only worry. These microplastics can also attract and carry other harmful substances. They're called co-pollutants. These might be chemicals added during manufacturing or just other pollutants that the microplastics sort of grab onto from the air, like how dust clings to surfaces.
So these tiny plastic bits become like little taxis for other potentially nasty chemicals floating around indoors. Okay. Now, how have we traditionally kept tabs on the air quality inside buildings? What are the standard methods?
Well, traditionally, monitoring has involved actively sampling the air, using pumps and filters to measure things like gases, larger particulate matter, and just general dust levels. But there's a growing interest in using what are called bioindicators, basically living organisms like certain plants or lichens or mosses, to give us a maybe more holistic view of air quality over time.
Okay, so this is where our eight-legged architects come in. Why spiderwebs? What makes them a good candidate for this kind of biomonitoring?
This is where it gets really cool, I think. Spiderwebs have several characteristics that make them surprisingly well suited for this. For one, they're practically ubiquitous. I mean, you can find them in almost any indoor space, right?
Sure, yeah.
And second, they naturally act like sticky traps. They capture airborne particles. That's their job in a way.
Yeah.
Collecting them is also pretty simple. It's cost-effective. And it doesn't really disturb the environment much. Plus, you often find them in these out-of-the-way spots where dust might not be regularly cleaned.
That makes a lot of sense. They're just there, doing their thing, passively collecting whatever's floating by.
The study also mentions they can help with something called temporal assessment. What's that about?
Exactly. So, because spiders often build new webs, researchers can, say, remove an older web and then study what accumulates in a freshly spun one over a specific period. This allows them to see how pollution levels might be changing over time. And while previous studies have looked at webs for outdoor pollution, this research really focused specifically on indoor air.
Right. So the researchers wanted to see if these webs could reliably tell them not only how much microplastic was present, but also what kind of chemical pollutants were hitching a ride on them, specifically indoors. How did they go about testing this? What was the method? Okay, so the researchers collected both spiderweb samples. They specifically targeted the webs of falsidae spiders. Those are the ones that make the really thin, tangled webs you often see in corners.
Ah, the cellar spiders, yeah.
Right. And they also collected regular dust samples from the same locations for comparison. They gathered these from a really diverse range of indoor locations in Cochin City, India.
And it wasn't just homes, right? The study looked at quite a variety of places.
That's right. It was pretty comprehensive. Their sampling spots included paint shops, schools, colleges, medical shops, antique shops, tailoring shops, textile shops, home decor places, photostat shops, table mat shops, even facilities that collect plastic for recycling.
Wow, okay. That variety seems crucial to get a broad picture.
Absolutely. You want to capture different potential sources of indoor microplastics.
Okay, so they had their webs and their dust. What happened in the lab? How do they actually identify and count the MPs?
Well, the first step was to clean the samples up, essentially. They needed to get rid of any organic material, like insect bits or actual spider bits. They used hydrogen peroxide for that. Then, to separate the plastics from other inorganic stuff, they used a technique based on density separation with zinc chloride. Think of it like panning for gold, but for plastic.
Okay.
The lighter plastic particles float. This separated material was then filtered, and the tiny particles caught on the filter were examined under a stereo microscope to actually count the microplastics.
But just seeing them isn't enough, right? You need to know what kind of plastic you're dealing with.
Correct. That's crucial. So the suspected microplastic particles were then analyzed using two pretty sophisticated techniques: Raman spectroscopy and ATR-FTIR analysis. These methods basically work by shining specific types of light on the samples and analyzing how that light interacts with the material. Different plastics have unique spectral fingerprints, so you can identify the specific polymers.
That's some serious detective work on these tiny particles. Now, what about those co-pollutants, the BPAs and phthalates? How do they analyze those?
Right, the chemical hitchhikers. For that, they needed a different approach to make sure they didn't destroy the chemicals they were looking for. They first separated the microplastics from the web and dust samples again. Then, they used a process called sonication, which is essentially using high-frequency sound waves to help extract any absorbed chemicals from the plastic particles into a liquid solution, methanol and water in this case.
Okay, like shaking the chemicals loose with sound.
Exactly. This liquid extract was then concentrated and analyzed using a highly sensitive technique called LC-MSMS, liquid chromatography tandem mass spectrometry. This method can separate, identify, and quantify even tiny amounts of specific chemicals. They targeted bisphenol A, BPA, and various phthalate esters, those chemicals often used to make plastics flexible, and even their breakdown products, the monoesters.
All right, so after all this meticulous lab work, what were some of the main takeaways? What did they discover about where these microplastics were showing up?
Well, one of the most striking findings, I think, was that the spiderweb samples consistently contained much higher levels of microplastics compared to the dust samples collected from the same locations.
Really? How much higher?
We're talking orders of magnitude sometimes.
Yeah.
The range was something like 138 to over 33,000 MPs per gram of web versus 59 to about 9,300 MPs per gram in dust. So yeah, significantly more concentrated in the webs.
Wow, that's a huge difference. But did the patterns match up? Like, if a place had high MPs in dust, did it also have high MPs in the webs?
Interestingly, yes. Despite the difference in concentration, they found a strong positive correlation. The stats showed R equals 0.93, which is very strong between the levels in dust and webs. This really supports the idea that spiderwebs can act as reliable indicators or bioindicators of the overall indoor microplastic contamination levels.
Okay, so they mirror the general trend but accumulate more.
Exactly. Though it's also important that the distribution wasn't even. They found that MPs were unevenly spread in both webs and dust, suggesting there are contamination hotspots within these indoor environments. High variability, statistically speaking.
So not every corner of a room is equally polluted. That makes intuitive sense. Were there any particular types of businesses or locations that stood out as having these higher levels, these hotspots?
Absolutely. They saw significant spikes in MP levels in places like tailoring shops, textile shops, home decor shops, photostat shops, table mat shops, and those plastic collection units.
Okay, so places dealing with fabrics, plastics, electronics.
Precisely. It strongly suggests that the activities and materials in those environments, cutting fabric, releasing fibers, handling lots of plastic items, maybe dust from electronics, are major sources of indoor MPs. And actually, the highest levels overall in both the dust and the webs were found in the home decor shops.
Home decor, interesting. So why do you think the webs accumulate so much more than the dust? What's the theory there?
Well, the researchers think it's likely a combination of factors. A key one is probably the lifespan of the web indoors. Unlike dust, which gets disturbed, cleaned, resuspended by us moving around.
Right. Vacuumed up, hopefully.
Hopefully. Spider webs, especially from these falsity spiders, which can live a while and don't always dismantle webs, tend to just hang there undisturbed for much longer periods. This gives them more time to accumulate airborne particles like microplastics.
Makes sense. Longer exposure time.
Yeah. And also, the collection method itself might play a role. They noted collecting webs carefully with clean glass rods into glass vials is probably less prone to contamination compared to sweeping or brushing dust, which might pick up other debris or lose finer particles.
So the dust we might sweep up regularly could be just the tip of the iceberg, in a way. These undisturbed webs could be giving us a more concentrated picture of pollutants accumulating over time.
That seems to be what the data suggests, yes. A potentially more integrated sample over time.
Okay, so the webs are great collectors. What did they find out about what these microplastics were actually made of, the different types of plastic?
Using those spectroscopic techniques, Raman and ATRIR, they identified three main types of plastic polymers in the web samples. Polyethylene, PE.
Like plastic bags and bottles,
Exactly. Polypropylene, PP.
food containers, packaging film.
Right. And polystyrene, PS, which is often used in things like styrofoam or disposable cups. Interestingly, though, they found polyvinyl chloride, PVC, in the dust samples, but not in the web samples.
Huh. PDC and dust, but not webs. That's specific. Any ideas why that might be?
The researchers theorize it might come down to density. PE, PP, and PS are generally less dense plastics. They might stay airborne for longer, increasing their chances of getting caught in elevated spider webs. PVC, on the other hand, is denser. It might settle out of the air more quickly and therefore accumulate more in the settled dust on surfaces, rather than staying airborne long enough to reach the webs.
Ah, okay. That makes physical sense. Lighter stuff floats longer. What about the shapes and colors of these MPs?
They mostly found what are called secondary microplastics, things like fibers, films, and fragments in both the webs and the dust.
Meaning they broke off larger items rather than being manufactured small.
Generally, yes. This suggests they're likely coming from the breakdown and degradation of larger plastic items indoors. They also saw a variety of colors. Dark blue or black MPs were common, especially in webs. They thought this might be from things like degrading furniture, building materials, or maybe just aging colored plastics losing their pigment. Transparent or white MPs were also common, potentially from single-use plastics or maybe from photo-bleaching colors fading due to light exposure.
Okay, so we're finding these tiny plastic bits from various sources accumulating in webs and dust indoors. And you mentioned earlier they carry chemical hitchhikers. What does this all mean for us, for our health?
Yeah, this is really the crux of the issue, isn't it? Microplastics, as we said, can act as carriers for toxic substances. The study focused on BPA and phthalates, but potentially heavy metals too. This increases our risk of exposure through breathing them in, swallowing them with dust, or possibly even skin contact.
And the plastics themselves can be irritating.
Yes, the MPs themselves might cause respiratory or gastrointestinal irritation. And the even tinier ones, nanoplastics, which are harder to measure, raise concerns about potentially penetrating cells, causing oxidative stress or triggering immune responses. So it's this dual threat, the particle itself and the chemicals it's carrying, like little toxic delivery systems accumulating right in our living spaces.
And those co-pollutants, the chemicals they carry, what kinds of specific health risks are associated with them?
Well, substances like BPA and certain phthalates are known endocrine disruptors, meaning they can interfere with hormones. Other potential co-pollutants, like heavy metals, could lead to things like kidney damage, skeletal issues, cognitive impairments, or developmental problems. And certain groups are more vulnerable: children, pregnant women, the elderly. Children especially, because of their behavior, crawling, putting things in their mouths, they tend to have higher contact with an ingestion of dust.
Right. The study specifically tried to estimate the risk from ingesting MPs through dust, didn't it? What were those findings?
Yes, because there isn't really a standard method yet for assessing risk directly from MPs and spiderwebs. They focused on estimating the potential daily intake, the EDI, of larger MPs over 50 micrometers through unintentional dust ingestion. They found that those home decor shops had the highest estimated intake risk through dust. Antique shops and educational institutions were on the lower end.
And within schools?
Within schools, interestingly, the estimated daily intake for children was highest compared to teenagers and adults, likely reflecting those behavioral and physiological differences.
Definitely food for thought, especially for parents in schools.
Huh.
Okay, let's circle back to those co-pollutants, the chemicals on the plastics. What did the study find about their levels in the web MPs versus the dust MPs?
This is a really key part of the study and quite novel. They successfully detected BPA and several different phthalate esters, DBP, DEP, DMP, plus their breakdown products, the monoesters MBP, MEP, MMP, and the microplastics extracted from both the spiderwebs and the dust.
And finding them in the web MPs is a first.
Yes. According to the paper, this is the first time these specific organic co-pollutants have been detected and measured in microplastics collected from indoor spiderwebs. It confirms the webs aren't just trapping plastic, they're trapping the associated chemicals too.
So the webs are concentrating these potentially harmful chemical hitchhikers. Were there differences in the types or amounts found in web MPs versus dust MPs?
Yes, there were some quite interesting differences, actually. Overall, The total concentration of all the measured co-pollutants was higher in the microplastics from the webs than from the dust.
Higher in the webs again.
Higher in the webs again, yes. This was mainly driven by high levels of one specific phthalate, diethyl phthalate, or DEP, in the WebMPs.
Yeah.
However, when they looked at individual chemicals, the trends varied. DEP, DMP, and MEP were generally higher in WebMPs, but BPA, DBP, MBP, and MMP tended to be more concentrated in the dust MPs.
Huh. So it's not a simple more of everything in webs picture for the chemicals. DEP is high in webs, but BPA and DBP are higher in dust MPs. Any ideas why these specific differences might exist?
The researchers suggest these variations could be due to a whole range of complex factors specific to the web versus the dust environment. Things like physical factors, airflow patterns, surface properties of the web versus dust particles. Chemical factors, how strongly different pollutants bind to the plastic versus how readily they might partition back into the air or onto other surfaces.
Yeah.
Maybe even biological factors related to the spider or microbes present.
So the web and dust act as different kinds of chemical sinks or reservoirs, maybe?
That's a good way to put it. The partitioning coefficients, how chemicals distribute themselves between different phases like solid plastic, air, or surface dust, are likely different and play a big role.
And what about finding those breakdown products, the monophthalate esters? What's the significance of that?
Finding NBP, MEP, and MMP, the breakdown products of DBP, DEP, and DMP, is significant because it shows that the original phthalate chemicals are actually undergoing transformation, breaking down, within our indoor environments. It also suggests that microplastics can act as reservoirs, not just for the original pollutants, but for these transformation products as well. And this matters because sometimes these breakdown products can be more persistent, more reactive, or even more biologically available, potentially more harmful than the parent chemical.
That adds another layer of complexity. The study mentioned briefly that webs have been used before for other pollutants.
Yes, that's right. It's not a totally new idea to use webs. Previous research has shown they can be effective for monitoring things like airborne heavy metals, PAHs, polycyclic aromatic hydrocarbons, even dioxins, mostly in outdoor settings, though. This study really builds on that, showing their potential for MPs and these specific organic chemicals indoors. And given their stability compared to dust, their ability to integrate exposure over time, and how easy they are to collect non-invasively, they really do seem to hold promise for studying the fate and transformation of pollutants indoors.
Okay, so wrapping this up, what's the big picture message here? What does this all mean?
Well, I think this research really shines a light on this kind of hidden world of microplastic and chemical pollution that exists right inside our homes and workplaces, often unseen, but clearly present. And it highlights the really surprisingly effective role that something as commonplace as a spiderweb can play in trapping and concentrating these contaminants.
And the fact that these webs seem to accumulate higher concentrations, at least of the MPs overall, and certain co-pollutants like DEP compared to the dust we might sample.
Exactly. That suggests they could be an incredibly valuable, low-cost, and non-invasive tool. A new way to keep an eye on our indoor air quality and get a better handle on the potential risks we face. They might even give us a more accurate picture over time of chronic exposure compared to a snapshot dust sample.
And here's where it gets really interesting for me. Those differences they found, PVC only in dust, DEP high in webs, DBP high in dust MPs, that variation really hints at complex processes happening indoors. It suggests different pathways for how contaminants move and accumulate, and maybe even different chemical transformations happening on the web versus in the dust.
It absolutely does, which raises a really important question. If spiderwebs are such effective passive samplers, could we potentially use them more widely? Could we, say, map indoor pollution hotspots by collecting and analyzing webs from different homes, schools, offices? Could this give us a much better understanding of where these substances are coming from, how they move around, and where the risks might be highest?
You could almost imagine community science initiatives, people collecting webs carefully.
Potentially, yeah.
Yeah.
It opens up possibilities for much wider, lower-cost monitoring.
So for you, the learner, listening to this deep dive, hopefully it offers a completely new perspective on something you probably walk past every day, a spiderweb. It turns out these intricate little structures aren't just inert. They can offer really valuable insights into our health and the quality of the air we breathe indoors.
Yeah, so as you go about your day, maybe take a moment to actually notice any spiderwebs you encounter. They are silently, constantly collecting information about the air around you. And as this research shows, they might be telling a really important story about our indoor environment, one that we probably need to start paying more attention to.
And one final provocative thought for you to consider. If spider webs can reveal so much about microplastic and co-pollutant levels, what other unexpected natural or biological indicators might be out there? What other parts of nature, maybe even indoors, might be holding clues about the world around us, just waiting for us to figure out how to read them? Perhaps exploring the field of biomonitoring could uncover other surprising sentinels of our environment.